Biology is the study of living things. Evolution is basically the result of biology. In order to have evolution occur, you have to have cells. Cells are a phospholipid bilayer encapsulating a liquid called the cytoplasm. This liquid is mostly water, the same as cells have to be in a saline solution on the outside. Saline means it's salt water that's the same as inside the cell. Water is the reason things are alive, because water is a universal solvent of living things. Water is a polar molecule, and as a polar molecule, it's a very nice solvent. In fact, it is the only solvent known to support the complex molecular interactions of life. It has to be inside a cell for this to occur. Because if you had the chemical reactions occurring outside of the cell, they would wash away in the environment. So the cell is basically a reaction chamber in which the chemical reactions of life can occur. In this reaction chamber, you have the process of DNA transcribing information onto RNA. DNA and RNA are both made up of nucleotides, and these nucleotides can retain information. DNA is stable because it's a double molecule, and both of them are made out of two types of nucleotides. The purines are adenine and guanine, and they have two rings as well as the phosphate backbone and the sugar that is deoxyribose. That means that the sugar is what the nucleotide, the nitrogenous base is connected to, and the phosphate holds each one to get to the other, so it hooks together. With DNA, one strand will line up and it will cause the replication of itself, so it can carry the information from one strand to another strand, and then when they split, each strand can make another strand. This is called replication. The information that is taken off and put onto RNA can be decoded into proteins. Before it is decoded into proteins, it has to be manipulated in the nucleus and certain parts cut out and left behind. They're called introns. Enzymes do this. The exons are put back together, and the 5' and 3' end are added to them. After that, they can get out of the nucleus in eukaryotic cells and go to the ribosomes as messenger RNA. The nucleotide bases in units of 3, called codons, will code for specific amino acids that are brought in by another type of RNA called transfer RNA. The transfer RNA has the anticodon. It carries in the amino acid, and the ribosomes will be involved in exposing the messenger and connecting the amino acids together. Amino acids are what make up proteins, and they have a nitrogenous group on one end and an acid group on the other. The acid group is a carbon with a double bond oxygen and an OH group to it. Carbon will make 4 covalent bonds. That leaves 1 covalent bond because 2 of them are with oxygen and 1 with another oxygen, and the oxygen makes 2 covalent bonds, so that one has another one with a hydrogen, which it can give up easily. And this is why it's called acid. The other carbon bond is connected to another carbon, and that carbon is then connected to a nitrogen, and it is also connected to a R group or an R chain. And the R chains change, but the amino group the amino acid group is the same with all of the amino acids that are used to make proteins. Once these amino acids have been hooked together at the ribosome, they are called peptide bonds, and so when you have several of these connected, they're called polypeptides. Proteins are polypeptides. The proteins are the structural units of our bodies. That's what the cell basically is a protein factory, and we are made out of trillions of little tiny protein factories. They are based off of the code that is found on the DNA, and that code is like a program. It's like a computer program. So if you're using an Excel program, you can use it to do multiple different jobs. Well, this same program inside the cell can be used to code for multiple proteins. Consequently, one cell can use it to make one type, and another cell can use it to make another type. And there are several different programs on our DNA, obviously, since we have 23 sets of chromosomes, and each chromosome has multiple genes on it. We are able to make many different proteins, and the proteins that we make determine what we are. So the DNA makes us look like we are, and the DNA that is very similar in chimpanzees makes them look like they look, which is very similar to us. Dogs are much more distant, so they don't look as much like us because they have different proteins that are being manufactured due to mutations and things that have occurred along the way. The cell exports these proteins and also uses them for a variety of functions inside. As a result of the activities of the cells, we are built up into what we are. Consequently, what we really are is just a huge colony of individual living units, each one an individual life form of its own right. What we are, what we consider ourselves, is nothing but the colony that has been produced by all of these little individual factories combined together using energy that they got from the nutrients in the environment. One of the functions of life is the ability to acquire and use energy in order to grow and to reproduce. That's basically what the cell is. The next time I will go into something else. So for now, I will go ahead and stop if I can.